Uh, okay. Okay. At first, Professor Ski, thank you for introduction. I'm Lida from Computational Optics Groups, University of Tsukuba. Today, I'm going to talk about a multi-contrast tissue imaging with numerical refocus based on polarization-sensitive optical coherence tomography. Recently, with the development of tissue cultivation skills, more kinds of tissues can be cultivated in vitro, which greatly promote the development of tissue investigations such as drug response studies. For such a tissue investigation, it requires an imaging tool which is able for multi-contrast imaging, visualizing microstructure, and having a deep penetration, and also can monitor the dynamic activities. Conventional multi-contrast imaging methods such as staining and fluorescence microscopy can reveal properties or activities of the tissues. However, for multi-contrast tissue imaging, it also requires high penetration for a few millimeters and also a high resolution, at least few tens of microns, and it also needs to be non-invasive. But none of these methods can suffice all the requirements. Chemical staining and fluorescence microscope are invasive, and phase contrast microscopy has limited imaging depth with nearly 100 microns and surely not sufficient for thick tissues. Polarization-sensitive optical coherence tomography, also named as PSOCT, is a functional extension of OCT technique. It can non-invasively measure the intensity and the birefringence property of the sample and has a few millimeters of imaging depth. However, just like other methods, it suffers from a trade-off between the lateral resolution and imaging depth. One of the solutions to overcome this trade-off is termed as computational refocus. This is numerical processing based on a forward propagation model to achieve high lateral resolution along whole depth. Here are the examples of human anterior eye segments with defocus imaging. We can see the iris stroma becomes clearer after being refocused. However, this method has only very few implementations in PSOCT yet. Its application in PSOCT measurement has not been fully exploited. So, the purpose of this project is to use PSOCT to measure the tissue sample with multiple contrast and implement computational refocus to overcome the trade-off issue. In the experiment, we used a custom-built PSOCT system. It has a central wavelength of 1.3 micron, and the lateral and axial resolutions are 19 and 14 microns in tissue respectively. It has a passive polarization delay unit and polarization diversity detection unit to obtain the OCT signals from 4 different polarization channels and measure the birefringence of sample. And to each data volumes, the bulk phase error and jitter error are corrected by our self-development algorithms. After the phase is stabilized, unfast images at each depth are extracted for the upcoming computational refocusing. For example, for each depth, as demonstrated here, first only unfast image from single channel is extracted in Fourier transformed, then multiplied by a deconvolution phase filter, and then inversely Fourier transformed back. This filter is optimized by minimizing the information entropy of the refocused images along certain depth ranges. After the filter is determined, it's used for refocusing all channels simultaneously, and this process is extrapolated to all depths to refocus the entire data volumes. Finally, from the refocused data volumes, we can estimate the birefringence of samples by applying a maximum a posteriori estimation with noise correction. Next, I'll introduce the sample and the measurement protocol in an experiment. The sample was porcine triceps brachii muscle measured ex vivo. The scanning range was 2 by 2 mm square, sampled by 512 by 512 points, which corresponds to a lateral pixel separation of 3.9 microns. The total measurements took about 3 seconds. Here, I show the cross-sectional intensity image of the sample. On top is the original image without refocusing, and at the bottom is the one with refocusing. During the measurement, the focus was manually set beneath the sample surface. Here, a single depth outside the depth of focus is selected, denoted by this red line. You can see the original unfast image shows strong blurring, but after being refocused, the vertical aligned textures of muscle fibrils can be observed here. And also the edges around the central hollow region become sharper as indicated by these red marks. Here, a single line at the hollow regions is selected and the line profile is plotted. Blue and red lines correspond to the profiles without and with refocusing respectively. It shows the sharpness of the edge is improved by around 25%. And the result at other depths also show the similar resolution improvement. It's just that volumetric refocusing has been successfully applied over a millimeter. Next, I'll show the results of the birefringence images, which is composed with intensity and displayed in RGB color space. Here, the birefringence image, we can observe the fibril structures, which agrees with the intensity image, and it has relative high birefringence here in the image shown as green. It agrees with the fact that muscle fibrils consist of collagen, which is considered to exhibit birefringence. Another finding of the birefringence result is that in the original blood image, we can see some abnormal high birefringence as the yellow arrow indicates, but it decreases to normal level after being refocused. Here we can observe this green on the fiber and the blue on the surrounding connective tissue. Here, 4 regions of interest that exhibit this high birefringence signal were selected by using a window of 15 by 15 pixels and to calculate the average level of the birefringence. The results shows that the original birefringence level is higher than after applying computational refocus. And it also can be observed at other locations. Results show that the original data with low resolution mostly exhibits this high birefringence signal, and it can be found in other data. As for the source of this high birefringence, we propose 2 hypotheses. First, this is a true birefringence signal which correctly reflects the property of sample, but after applying computational refocus, it was falsely decreased. And second, this is a false birefringence signal that cannot represent the true tissue property, but it is corrected by computational refocus. Namely, it was caused by the original low resolution. So, to validate which one is correct, an additional experiment was performed. We measured the same sample twice with different focus positions so that the same depth can be imaged both outside focus and in focus. Later, by applying computational refocus or defocus, we can numerically shift the sample plane and change the solution. The original images can be treated as physically reliable references, so we will have 4 images with 2 different kinds of resolutions. Here are the results. The left column is the physically defocused image group. A is the original blood image, and C is the numerically refocused one. On the right column is the physically in-focus image group. Bottom is the original and top is the numerically defocused result. Namely, here in B and C are the numerically generated images. By comparing A and B, they all showed the similar high birefringence signals as the yellow arrows indicate, and in C and D, they're significantly suppressed, just as the previous results suggest. So, several areas out of interest where the high birefringence exists are selected, and the average birefringence are compared. First, let's compare the results with the same focus position. By comparing A and C, we can see the birefringence becomes higher after numerical defocusing. And also, the same between B and D, it agrees with previous conclusion. And next, let's compare the results with the same resolution. By comparing C and D, we can see that except for area 3, all the birefringence mostly agrees to each other. And also, the results of A and B shows the same consistence. It suggests that both the numerically refocused and defocused results mostly agrees with the physically imaged references and thus is reliable. Now let's look back at these 2 hypotheses. The first one says this high birefringence signal is true but decreased by refocus, which means it should exist in physically but instead of numerically in-focusing image. As a result, numerically in-focusing image should have lower birefringence than physically in-focusing image. Similarly, in the second hypothesis, if it's artifact but corrected by refocus, as a result, the physically in-focusing image should have lower birefringence than numerically defocused one. The facts from previous experiments are listed here. Obviously, it violates the first hypothesis and therefore is not correct. And the second hypothesis can explain the results, so the conclusion would be this abnormal high birefringence is most likely to be an artifact that caused by the low resolution, which can easily happen for thick tissue imaging if the trade-off is not overcome. So now let me conclude my talk. In summary, we demonstrate this application of computational refocus using polarization sensitive OCT system to solve the trade-off and achieve imaging depth of a millimeter with high resolution. And in intensity and birefringence images of muscle samples are shown, and the results show that shows clear fiber structures where exhibit birefringence. And in addition, some high birefringence artifact is observed under low resolution, which can be significantly suppressed by applying computational refocusing. So based on these results, we conclude that computational refocus is important for quantitative polarization evaluation for tissue imaging. Yeah, so basically, that's my talk, and here I would like to thank my COG colleagues and co-authors, and thank you for your attention.